Hey guys, welcome back to my channel. So after a long time I am uploading the video. So today I'm going to start with a new subject that is for pharmaceutical biotechnology for the 6th semester as per our GHS question bank. So I'll start from the 10 marks for pharmaceutical biotechnology. So the first question for the 10 marks, that is long as they explain the different methods of immobilization of enzymes and give its application. So the different methods for immobilization of enzymes are adsorption, or it is also known as a carrier binding method. Number second, covalent bonding. Number third, entrapment method. Fourth, copolymerization, and lastly fifth one, that is encapsulation. So we'll see the first one, that is adsorption or carrier binding method. So in adsorption, the enzyme is absorbed to external surface which supports the adsorption of enzymes onto the insoluble supports is a very simple method for wide applicability and capability of high enzyme loading. So the simple mixing is the enzyme with the suitable adsorbent under the appropriate condition for pH and iconate strength. So the after sufficient incubation period, by washing off loosely bond and unbounded enzymes will produce the immobilized enzyme in the directly usable form. So they also supports the carrier, which is used maybe in the different types, and in that the first one, mineral support, example aluminium oxide and clay. Organic support, that is starch and modify separates and ion exchange resins. So these, there are no permanent bond for the formation between carrier and the enzyme in the adsorption method. Only weak bonds were stabilized by the enzyme to the support or the carrier. So the weak bonds involves mainly ionic interaction, hydrogen bonding, and vulnerable forces. So in that, the method for the absorption are static forces process, where this is most efficient technique but requires maximum time for the immobilization to carry out along the following solution, where containing the enzymes to contact the carrier without stirring, and in that the dynamic batch process, where the carrier is placed on the enzyme solution and mixed by stirring or mechanical agitation, and in that number third, that is reactor loading process, where the carrier is placed in a reactor and then the enzyme solution is transferred to the reactor with a continuous agitation. And fourth one, that is electrode process, where the carrier placed near the electrode in an enzyme, where the bath then current is put on under the electric field, where the enzymes migrates to the carrier and deposit on its surface. So number second, that is covalent bonding. So the covalent bonding is the method which involves the formation of covalent bonds between the chemical groups in the enzymes to the chemical groups on the support or the carriers. So it is one of the most widely used method for the enzyme immobilizations, where the methods for the covalent bonding is azotization, that is bonding between the amino group of the support, where the example is amino benzyl cellulose of the enzyme, etc. And that B, peptide bound, where the bond formation between the amino or carboxylic group of the support and the amino of the carboxylic group for the enzyme. Group activation, where uses the cyanogen bromide to support the containing glycol group, that is cellulose, saccharose, etc. And polyfunctional reagent, which is used by the for the bifunctional or the multifunctional reagents, which forms a covalent bonds between the amino group to support and amino group to enzyme, works as a bridge between the enzymes and the carriers. And in that number third, entrapped method, entrapped, which interrupt the methods. So in these methods, the enzymes are physically entrapped inside the porous matrix bonds, which are stabilizing the enzyme to the matrix, may be covalent or non-covalent bonds. So these matrix will be used be a water soluble polymer to form a nature or a matrix which varies with the different enzymes. So pore size for the matrix is adjusted with the concentration of the polymer to prevent the loss of enzyme, where the entrapment is the method of choice for the immobilization of microbial, animal and plant cells, where inclusion in the gels, enzymes trapped within the gels, inclusions in fibers where the enzyme supported on the fibers made of matrix material, where inclusion on microcapsules where the enzymes entrapped in the microcapsule that forms by the monomer matrices such as polyamide and calcium alginate, where the fourth method for that is copolymerization or cross-linking. So this is also known as a cross-linking copolymerization. So in this method of immobilization, enzyme are directly linked by the covalent bonds between the various groups of enzymes via polyfunctional reagents, where unlike our other methods there is no matrix or support involved in this method, commonly used as polyfunctional reagents as well as a glutyl aldehyde and diazonium salt. And number fifth, that is encapsulation. So this is the immobilization which is done by enclosing the enzymes in the membrane capsule, where the capsules will made up of a semi-permeable membrane like nitrocellular or nylon. So in this method, the effectiveness depends upon the stability of the enzymes inside the capsule, where the applications for this are large quantity of enzymes can be immobilized by capsulation and waste water treatment in biodiesel production, and it is also applicable in the cheap and simple method, where the question number 2, that is describe the production of hepatitis B vaccine. So the hepatitis B virus is one of the most common infectious diseases to man. The World Health Organization estimated that were 285 million chronic carriers of these viruses in the worldwide. So hepatitis B is 50 to 100 times more infectious than AIDS. That is, hepatitis B results irritation and swelling of the liver due to the infection by hepatitis B virus. So hepatitis B recombinant vaccine production, where the nucleic acid and the hepatitis B has been identified as a 422 nanometer particles, where containing the double standard circular DNA molecule for the about 3 kb in the size, where the plasm is infected by the humans has been activated or deactivated to having the varying amount of hepatitis B antigens. So there are 3 types of viral coded proteins that are recognized by the antigenic, where in that number 1, viral surface antigen, that is HBS. HBSS indicates surface. So viral surface antigen, viral core antigen, that is HBC. So C indicates the core, and E antigen HBE. So E indicates E antigen. Recently HBSA antigen, that is HBS, that is viral surface antigen or its subunits, are useful for the production of recombinant hepatitis B vaccines. So after the infection in the human beings, the hepatitis B virus fails to multiply and infect the large number of cells and even does not grow in cultured cells. So using recombinant DNA technology, the vaccine uses the surface antigen, that is HBS antigen. So surface antigen of the virus that stimulates the production of protective antibodies which combat with the hepatitis B viruses. So the steps involved in the recombinant hepatitis B vaccine production. So HBS, that is surface antigen, producing a gene which is isolated from the hepatitis B virus by a normal isolation products like cellulose, protein denaturation, precipitation, centrifugation and drying method, where a plasmid DNA is extracted from the bacterium E. coli and is cut by the restriction enzyme by E. coli, forming the plasmid vector. And deisolated HBS antigen, that is surface antigen producing the gene, is located and inserted into the bacterial plasmic vector on forming a recombinant DNA, and these recombinant DNA containing the target gene is introduced into a yeast cell, forming the recombinant yeast cell, where the recombinant yeast cell multiplies in the fermentation tank and produces the HBS antigen, that is surface antigen. So the fermentation process involves in the growth of S. cerevisiae on the complexes fermentation medium, which consists of the extract of yeast, soy peptone and dextro amino acids, and as well as mineral salts. So after 48 hours, the yeast cells are ruptured to free HBS, that is surface antigen, and the mixture is proceed for extraction. That the HBS antigen protein is released from the yeast cell by desorption and purified by the series of physical and chemical method. So the purified protein is treated in the phosphate buffer with formaldehyde and then co-precipitated with alum, that is potassium aluminium sulfate, to form a bulk advocate with amorphous aluminium hydroxy phosphate sulfate. Each dose contains less than 1% of yeast protein. And then lastly, HBS antigen, that is surface antigen, are combined with preserving antigen agents, that other ingredients are sealed in a glass bottle. And where question number 3, that is explain the production of greasofulan and penicillin by fermentation technology with a neat label flow chart. So the production for the greasofulan fermentation technology, where the fermentation processes utilizes the microorganisms to convert the solid or liquid substances into the various products. So the fermentation is classified commercially either solid state or submerged fermentation. So the compound that is insoluble in water and slightly soluble in ethanol, methanol, acetone, benzene and ethyl acetate and acetic acid, etc., where the preparation for the fermentation of greasofulan is medium, that is Spazix-Dox medium, and the chemicals which are used for the preparation of greasofulan chemical is glucose, sodium nitrate, potassium hydrogen phosphate and magnesium sulfate, that is 7H2O water. So the steps involved in the manufacturing process for greasofulan is the fermentation, pre-treatment for fermentation of broth, filtration, extraction, decolorization, isolation and separation, precipitation and purification, where the fermentation for pH of the medium were adjusted between 6 to 7.2 medium, and the medium was dispensed in the fermentation with a fresh sample of mycelium suspension of fungus Penicillium crassophyllum from the fresh slope of the wrapper steep agar was obtained. So the solution were autoclaved for 200 minutes at 120 degrees Celsius with 15 lbs pressure and fermented for 14 days at 24 degrees Celsius. For the pre-treatment and fermentation broth, the broth is heated above 60 degrees for 20 to 30 minutes, and after heating, the sufficient coagulation of material occurs to produce a valuable improvement in separation that is characterized for the broth, where the period of heating may be short 5 to 10 minutes at the 80 degrees Celsius, having to be found provide a satisfactory increases in the filtration rate. And then the filtration method occurred due to the after the pre-treatment and the filtration of the fermentation, where the production for the penicillin by fermentation technology, where the fermentative production of penicillin, growth condition for the organisms, and even culture medium for growth of organisms consist, where the preparation of the penicillin fermentation is glucose or glycerol, polypeptone, yeast extract, and K2HPO4, magnesium phosphate with 7 water. So before use, pH of the medium was adjusted to 9 with 10% of sodium bicarbonate, which is sterilized separately. The optimum temperature for the growth of organism is adjusted to 30 to 37 degree Celsius, where the production for the penicillin fermentation is 300 ml flask containing 100 ml of culture medium, where the strain is cultivated at 30 degree for 23 hours on the rotary shaker, where the 19 hours after the inoculation, the 10,000 units of benzyl penicillase is added. Then inducer for the penicillase and cultivation was further continued for the 4 hours. Then the cell growth was followed by measuring the optical density, that is 610 nanometer. The most suitable sugar for enzymes production were glycerol and the concentration of 2 to 0.5 weight by volume, where the glucose at the concentration of 0.2% also enhanced the considerably of enzyme production, and the subsequent experiments for 0.2 was adjusted and to 0.9.0, where the enzymes were incubated by the penicillin and the cyclophosphamide derivatives. The amount of the enzyme produced reached maximum at 5 hours after the incubation of benzyl penicillin. If at any time the during growth period cell lysis were absorbed, penicillin should be added for 24 hours, or it is usually to be observed by the lysis culture that the enzyme filter the drops. And the fourth question, what are biosensors? Explain the types with the pharmaceutical applications. So what do you mean by biosensor? So the biosensor word is coined by the common according to the IUPAC recommendation. So a biosensor is an independently integrated receptor transducer device which is capable for providing a selective quantitative or a semi-quantitative analytical information using a biological recognition element, where it is essentially an analytical device which incorporates a biological derived recognition element to detect a specific bioanalyte integrated with the transducer to convert a biological signal into an electrical signal. So the types for the biosensors are their enzyme biosensor, where the first enzyme based on the sensor was reported by Updike and Hicks in 1967, where the enzyme biosensor have been devised on immobilization method. So that is the adsorption of the enzyme by van der Waals forces, ionic bonding or covalent bonding. So the commonly used enzymes for this purpose of oxidoreductase, polyphenol oxidase, peroxidase and amino oxidase. Number second, whole cell biosensor. So the first microbe based or the cell based sensor was designed by the devices. The tissues for the based on sensor arises from the plant and animal sources, where it is developed by the first tissue based on the sources for the determination of the amino acids or arginines. And number third, that is immunosensors, where the immunosensor were established on the fact that the antibodies have high affinity towards their respective antigens, that is the antibody specifically binds the pathogen to the toxins or the interact with the components of the host immune system. And number 4, that is DNA biosensor, where the biosensor was devised on the property that is single strand nucleic acid molecule is able to recognize and bind to its complementary strand in a sample, where the interaction is due to form an action of stable hydrogen bonds between two nucleic acid strands. Where number 5, the magnetic biosensor, which is miniaturized biosensor detecting the magnetic micro and nanoparticles in microfluidics channel, were using for the magnostereoism effect, have great potential in terms of sensitivity and as well as size. And lastly, number 6, the thermal biosensor or colorimetric biosensor, which are developed by the assisting biosensor material are in a physical transducer. Where number 7, that is piezoelectric biosensor. These are two types: quartz crystal micro balance and the surface of the acoustic wave devices, and optical biosensor, which consists of a light sources as well as numerous optical components to generate a light beam with a specific characteristics, and modifying the sensing with halong and photoelectric optical biosensor were devices that are utilized, where the principle for optical measurement optical biosensors primarily involved in the bioenzymes and antibodies were the transducing element. Optical biosensors are allowed to safe non-electrical remote using a sensing of materials, so they do not require any references for the sensors, as well as comparative signal can be generated by using the same source of light as well as the sampling sensor. And ninth, that is amphoteric biosensor. It measures either the current or the potential resulting from the chemical reaction. And 10th one, that is potentiometric biosensor, where it can detect the potential from the chemical reaction or electroactive materials, where when constant current is applied, the potentiometric biosensor can measure species such as pH, hydrogen, nitrogen, and other ions in the penicillin, etc. A good example, that is ISFET biosensor, were used to monitor the extramyocardial pH during an open heart surgery. And 11th, that is conductometric biosensor. So there are several reactions in the biological system that brings about the changes in the ionic species. So these ionic species alter the electrical conductivity, which can be measured. A good example. And lastly, applications for biosensors. So biosensors are in many fields, namely food industry, medical field, and marine sector, etc. They are provide a better stability in the sensitivity as compared to the traditional methods. So in food processing, monitoring food authenticity, quality, and safety. So the detection of aging of beer using enzymatic biosensor. Biosensors are used to determine the quality of the food, and it is also applied in the dairy industry. In fermentation processes, so the biosensors can be utilized to monitor the presence of production of biomass, enzymes, antibody. Electrochemical detection has been used to detect in the fermentation process. And number C, that is environmental field. Fourth, or D, that is medical field or biodefense, biosensing application. So the biosensors can be used for the military purpose at the time of biological attacks. And lastly, question number 5, write the detail on cloning vectors. So the cloning vector is a DNA molecule which is used in the vehicle to artificially carrying a foreign genetic material into the another cell, where it can be replicated with and expressed. A vector containing foreign DNA is termed as a recombinant DNA. So the DNA molecule used for carrying an exogenous DNA into a host organism and facilitates the stable irrigation and replication inside the host system is termed as a vector, where the characteristics for the vectors, it is capable for replicating inside the host. ROP, that is replication of protein or proton, then have a selectable marker for screening of recombinant organism, having a selectable marker and restriction for the site of the endonucleus, which is cut. Small size of vector is very beneficial to be handled, and high copy number, where the cloning vector are the DNA molecule that can carry a foreign DNA segment and replicate inside the host cell, where the most commonly used cloning vector are a plasmid or a PUC. The vectors which are used as a cloning vector, those are plasmid, bacteriophages, cosmids, YAC or DAC. In that the plasmid, so the plasmid are the extra chromosomal self-replicating and double-stranded closed and cellular DNA molecules usually present in the prokaryotic cells, and then they carry a foreign DNA with 15 kbp size of the bacteria. And in that PUC cloning vector, that is plasmid, University of California, where the plasmid are small, high copy number. Plasmid of the size is 268 6 bp, where the PUC vectors contain lag sequences and the gene multiply cloning site, where MCS with LAS. This helps use in broad spectrum for the restriction endonucleus and permit the rapid visual detection of an insert. And number second, that is bacteriophages. So these phage are the viruses that infect the bacterial cells. Some phages are widely used as a cloning vectors because the small size of the cloning were largely the plasmid are not suitable as larger inserts the plasmid sizes. So the phage has a linear DNA molecule which will produce two fragments following a single break. And number third, that is cosmid as a vector. So the cosmids are the plasmid that incorporate a segment of the lambda bacteriophage. Cosmid vector are prepared by inserting the cos sequences of a phage into a small plasmid vector, where the YAC, that is YAC, yeast artificial chromosome vectors. So the concentration for YAC provides more advantages over cloning and the bacterial cells. It can clone very large of DNA fragments. And this is bacterial artificial chromosomes vectors. So the BAC involves a cloning system that is derived from the particular plasmid found in the bacterium E. coli. So the use of the BAC allows a large piece of DNA bacterial or the non-bacterial sources to be expressed in the E. coli, where the process called the conjugation. In conjugation, two E. coli bacteria can physically connect and an exchange of the DNA can occur. So here we have completed the 10 marks question and answers for pharmaceutical biotechnology for 6th semester. So tomorrow I am going to continue with this. Please do like, share and subscribe to my channel.